podcast pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today. In this episode, we take a look at how bioengineering can enhance delivery of essential drugs, how it can help with building artificial organs, and even determine which anti-cancer drugs are most effective in an individual. My guest today has helped to guide such research for the last 4 decades. Professor Robert Langer leads one of the largest bioengineering labs in the world at MIT, where he has pioneered the fields of controlled drug release and tissue engineering. We also discuss the challenges of attracting funding in science and deciding which projects are worth pursuing. So, Professor Langer, welcome. It's a pleasure to have you with me today.
Well, pleasure talking to you. Thank you.
So your lab works, in its own words, at the interface between biology and material science. And I was wondering, could you explain a bit more about what that means today?
Sure. Well, I mean, there's so many ways that materials make a contribution to medicine and other things. But I mean, for example, this whole area of biomaterials, any type of medical device, like a stent, pacemaker, artificial kidney, those are all biomaterials are central to them. Then there's a whole area that we work on, like drug delivery systems, which also can involve implants, including microspheres and nanospheres, which are critical for, like, say, gene therapy and things like that, the delivery of those agents. And then there's newer areas, like the whole area of regenerative medicine and tissue engineering is largely based on materials and biology too, and many others. I think it ends up being an incredibly, broad area.
And touching on the idea of a broad area, it's often said that experts are knowing more and more about less and less today. So does leading such a large group in Boston, Massachusetts, help with such collaboration?
Well, it is, but also our group is very interdisciplinary. We probably have about 11 or 12 different disciplines in our lab alone, and that makes a big difference because we have just all the people in our lab that can do everything from early stage biology, molecular biology, or chemistry to human clinical trials. So it's great having such a diverse group.
And moving on to drug delivery, so if you could sum it up, how can improved delivery make a drug more effective then?
Well, I think there's 2 things. First, it can make it more effective because if you keep it at the right rate, you won't have side effects or you'll just reduce them. Secondly, it enables you to improve, like a lot of people forget to take their drugs, and that leads to hospitalizations and side effects and sometimes death. I think the New York Times estimated that $300 million of healthcare costs in the $300 billion, of healthcare costs in the United States alone are due to people not taking their medicine. And then I think, most importantly for the future, it enables, it's not just improves therapy, it enables therapy. Like there's new drugs like siRNA and messenger RNA and, different types of gene editing. And really, I think most people feel the biggest challenge is delivery. And as an example, the first siRNA, this is a way to shut a gene down. This is a drug that ever got approved. This was Bioengineering last year. That involved using a nanoparticle to get it to the right spot, and to shut the genes down. So that was really important. And so I think we'll continue to see those kinds of things be very, very important.
And almost on the flip side, do you see a new life for drugs that perhaps previously failed clinical trials or preclinical trials actually being transformed into a new effective agent?
Yes. So, I think that's already happening. Different people have taken drugs and made them work better by drug delivery. So absolutely, I think one example, there was a cardiovascular drug, and it was used to be given by some type of gelatin capsule underneath the tongue, and then they put it in a controlled release osmotic pill, and it got approved for new indications like.
I know you mentioned controlled
congestive
release.
heart failure, I think it was, actually. Yeah.
Okay. And I know you mentioned controlled release as one example of perhaps a new way of optimizing drug delivery, but are there any other projects that you are working on currently in that sphere?
We have a lot. Let's see, where should I start? Well, we're working on new pills you could swallow that could last for weeks to months to years. We're working on new pills you could swallow that could deliver large molecules like insulin or genes. We're working on pills that are smart, that could have sensors in them. We're working on implants, like little microchips that could be smart and You could do remote control drug delivery. We're also working on injections that you could do for vaccines by a new approach that we developed, where hopefully you could just give one shot to babies for their entire life. We are working on nanoparticles for delivery of messenger RNA and DNA and siRNA. So there's a whole bunch of different things like that. And then in tissue engineering, we have, we've come up with new ways to hopefully treat people with hearing loss, and we're doing work on spinal cord repair, new treatments for new artificial pancreases for diabetes. I mean, new methods of giving nutrition to people in the developing world. So there are a lot.
And I'll move on to regenerative medicine a bit later on. But just following on from what you said, do you think that you're more focused on developing delivery mechanisms for drugs in general or for particular drugs? Do you say, actually, we'd like to deliver this drug, or we'd like to develop something that's kind of universal for a class of drugs, for example?
Well, I think generally what we've done is we do things that are kind of universal, but we can apply them to specific problems, and we do that.
And I think from that kind of long list of different potential projects that you're working on currently, another thing struck me, which is safety. How do you go about testing the safety of these delivery mechanisms, and do drugs have to undergo further trials?
They usually do. Yeah, so they usually do. So, we do sort of standard tests. We would look at safety for the drugs by liver function studies, blood tests, pathology, histology, looking at whether there's any tissue or organ damage. So I think the tests, carcinogenicity, I mean, a lot of them have been laid out by us and others in the past, and, they're used as a basis for FDA approval for different things.
So these are not replications of the clinical trials initially for these drugs?
Well, you start out with tissue culture things and animal studies, and then you go to, then you keep going up the ladder, more sophisticated animals, and then or higher level animals, and then human beings.
And do modifying the drugs themselves and developing different delivery mechanisms, such as controlled release, walk hand in hand? In your view, are you involved in actually altering the drugs that you're delivering as well to fit your delivery mechanisms, or is it working in the other direction mainly?
Well, we do both. We go in both directions. I mean, sometimes we've been given problems and we figure out the right delivery systems. Other times there's been interest in particular kinds of drugs, and so it varies. We do both.
And now, if I may, if we upgrade from molecules to something slightly larger, I know that you've been involved in work to develop polymer capsules for pancreatic beta cells. So how did this project come about?
Well, actually, that's a good question. That actually came about because one of the women, Julia Greenstein at the Juvenile Diabetes Foundation, called me up and said, that's the biggest limitation to making an artificial pancreas. People have worked on that and spent billions of dollars for over 30 years, and they'd never solved the problem, and could we help them on that? And so they gave us, myself and Dan Anderson, who was my postdoc then, now he's a professor at MIT, funding. They still do. But we've made, I think, some really substantial progress, and certainly have shown that we can get very biocompatible capsules in non-human primates.
Sorry, so what exactly was the problem that you started off with then?
Right, so the issue that, the problem in making the capsules is that whenever anybody did this, they always got fibrous encapsulation. In other words, they got scar tissue and other things around the capsule. And the problem with that is the artificial pancreas, you need to put it in a capsule so that it won't be rejected by the immune system. But then what happens is when you put it in the capsules themselves get all this fibrosis and scar tissue. So we needed to come up for the first time with a method or a material that wouldn't get fibrosed. And like I say, at least in non-human primates, we've been able to do that.
So how exactly did you manage to do that then in your lab?
Well, we did several things. First, there's always two things that you need to do. I should take a step back. What we proposed is this idea of high-throughput synthesis and high-throughput testing. So rather than just test one molecule, we thought we'd look at thousands. And so we did. We came up with some chemistries that would enable us to synthesize thousands of these molecules, of these substances. And then we came up with much faster ways of evaluating them in vitro and then even in animal models. And that enabled us to find some materials that were extremely biocompatible. And then we tested them in non-human, in a variety of animal models, including non-human primates.
And I think when I hear the numbers in the thousands, right now kind of the buzzword that I come to is artificial intelligence and machine learning. So are you using any of those techniques in your
We're starting to do
now?
that. We've had a number of talks with people that do artificial intelligence at MIT. So those are absolutely technologies that we're exploring.
So we're talking about a capsule for pancreatic beta cells, but could we go one step further and actually have scaffolds for kind of growing organs entirely for transplantation outside the field of diabetes?
Yes, we're doing that. Yeah.
And what do you think the barriers currently to that are?
Well, I think the barriers are immune rejection, which this tries to address. Getting enough cells, getting a vascular system, sometimes getting innervation. It depends on the tissue or organ you're trying to work on.
And have you learned any lessons from kind of research into tissue engineering that's perhaps fed into drug delivery as well, or vice versa?
Well, there's a lot of synergy between the two. In fact, you could use those capsules by, for example, and we're doing that. Like you could take a cell, put it in one of those capsules, but you could genetically engineer it so that it would make whatever drug you want too, like a protein, like erythropoietin. But yeah, there's lots of synergy and back and forth. And of course, making something biocompatible, that's good for everything that you put in the body, drug delivery systems, tissue engineered implants, but everything, because fibrosis is it's kind of a universal problem.
It's a major issue, of course. So do you see your lab playing a role, therefore, in the precision medicine era, in which treatments are targeted to individual patients?
Yeah, well, we've also developed an approach on that I'm excited about. Ali Jonas, who was one of my postdocs, he's a professor now. So he did a lot of work on this. And basically the way it started is Michael Sema, one of my colleagues, and I and some of our students, like John Santini, developed these little microchips that could literally deliver hundreds of drugs or hundreds of doses of the same drug. But what Ali did is he designed a little system based on that and other things that you could do for cancer therapy. And I'll just go over it right now. You may be familiar with the fact that a lot of times that people are thinking about personalized medicine, they actually take the tumor of the patient out and then they grow it in a dish and then they hit it with maybe 100 different chemicals.
A so-called N of one clinical trial.
Right, and that might dictate to you, what drug to use for that person's tumor. But when you do that, even though I think that's a good approach, it misses certain things, right? It misses the immune system, it misses the person's blood vessels, it misses the extracellular matrix. So what we did is rather than take the tumor and bring it to the laboratory and test it, we're actually bringing the laboratory into the tumor and testing it. And the way we do that is with Ollie's little device, like a kind of microchip, you can put 40 or 50 different drugs in it, and you can go in. And so what we do is when this patient has a tumor, and this is now in different clinical trials, but it worked very well in animals. What you do is when the patient has the tumor, you go in and you have a little biopsy needle, and the device is small enough that it fits in that biopsy needle, and you inject a few of them into the tumor, and then you come back, and they will release the 40 or so drugs, and then you come back a day later, and you take that needle out, the little device out with a slightly bigger needle, and a little of the tissue with it, and then you analyze the tissue, with the tissues adjacent to where the different drugs were by apoptosis or whatever, and it starts to tell you, well, which drugs work best on that patient's tumor, and that starts to dictate, well, what drugs or drug combinations might you use.
Wow, that sounds amazing. And could you see this method being applied beyond chemotherapy, for example?
Yes, we've talked about that too. I think anytime you had a disease that, might be useful, for example, in the brain for someday dictating drugs that might be useful in treating brain diseases, but I absolutely, you could envision it anywhere.
The brain is certainly occupying a very privileged position in relation to the bloodstream. It's been quite difficult to access it with therapeutic agents.
Also true, yes.
Have you made any progress, or has your lab been working at all into enhancing drug access for central nervous system conditions?
We've done it by implants. Like, for example, Henry Brem and I, we developed in our lab new polymers, polyanhydrides, and then working with neurosurgeons at Johns Hopkins, we came up with these little wafers and these wafers can locally deliver the drug to a brain tumor. They're called gliadel. So those have actually been in clinical practice for 23 years in over 30 countries now.
I might move on now to some more broad questions on the scientific world in general, if that's okay.
Of course, anything you want.
And I think nearly 300,000 citations later, and heading up one of the largest biomedical engineering labs in the world, if not the largest, I feel I can talk about another question. And I think I'm right in saying that you had your first 9 scientific grants turned down?
Yes, I got a very rocky start.
So I was wondering what your thoughts were on the role of failure in shaping scientific careers.
Well, I'd rather not have had failure, but I think
Because recent research has suggested that actually missing out on early grants actually correlates with a higher impact in the future. Sorry, failure is probably the wrong word to use.
Well, no, it's not. It's not. I mean, failure, but a lot of times if somebody does something very different, and I certainly did at the time, I was like one of the very few engineers, maybe the only engineer working in the medical field doing a lot of experimental work. I think that people were very skeptical of that.
And do you think that there are good and bad ways to approach kind of career offsets like that? If you were to give advice to people at this stage now.
I don't know that there's any good ways. I think that it's kind of like the road less taken. I mean, to me, it's a gamble. I've always been a little bit of a gambler. So, I wish there were easier for people, but I do feel, working in good labs, I was very lucky, I feel, to work in Judah Folkman's lab as a postdoc, and that was really important.
Even now, how do you go about putting projects to one side and maybe moving on from them if they're not being fruitful in the moment? Because that must be quite difficult, especially if a lot of resources have been put into them.
Yeah. So, well, I'm trying to think. I don't give up very easily, so we keep trying. In terms of giving up, I mean, I guess if I really felt there was a really good reason for giving up, I would. In other words, if one time, for example, we were developing a new treatment with immobilized enzymes for treating people with bilirubinemia. They had jaundice, and so we came up with a good paper, a good idea. It was using enzymes, and it worked in animals. But then people came up with a better idea around the same time about using light, and so because they had a better idea, there didn't seem to me any reason to push ours. I mean, on the other hand, a lot of things that we've done have taken a long time. And like when we were talking about that capsule device for diabetes, I have to give the Juvenile Diabetes Foundation that funded us a lot of credit, because the first 7 years we published no papers. But then in the last couple of years, we published like 8 papers in different nature journals, really not only finding these molecules that I mentioned, but also elucidating the mechanism and the right locations and all kinds of things. And research takes a long time, and sometimes there's no other way than to just keep at it and keep working hard.
And I know that you were asked in 2014 what you thought the biggest challenge facing scientists then was, and you said funding. Do you think that this is still the case?
Yep, I do. I think that, especially funding for basic research, but I think funding for any research is difficult. And so I do think that is, and especially for young scientists, funding is still, I think, the biggest challenge, because a lot of people have really good ideas. So sometimes people may have great ideas, but they're not necessarily great grantsmanship people. But I do feel that that's the biggest challenge still.
I think for scientists at the beginning of their careers, they might not necessarily afford the same kind of luxury.
They don't have a track. Yeah, they don't have a track record, and I mean, and now even with the government in the U.S., I think that you can't get something like more than 3 grants or $1 million, and I think with grants, one of the other problems is if you are somewhat different, like I was, or you come up with very strange ideas, the people who review the grants are often going to reject them. So, I mean, that's why I got mine rejected, because I wasn't, I was trying to do cancer research and biomedical research, and I wasn't, an MD, and I didn't know any biology either.
So I imagine the Langen Lab now has slightly more sway in if it doesn't publish for several years, or if it presents something slightly.
We do, but everybody, still depends on the place. If we got reviewed by a academic study, I mean, a place like Bill Gates, who funds us, or Juvenile Diabetes Foundation, they may not care. They have a different criteria. They want to see new treatments for diseases or helping people in the poor world, depending on which one. But it's the academic groups they'll look at publications, and I'm not sure that that's such a bad thing.
And you said that's not such a bad thing, because obviously publications are one way of looking at.
Yes.
It's a relatively easy kind of criteria to use for scientific success. But do you think they always show the entire picture?
Well, they don't, but the problem is figuring out a better way. I mean, I'd still rather it be based on a critical scientific review of which papers are going to be a part, rather than some bit political thing, where that somebody knows somebody and they'll give them a grant. I'd like it to be as merit-based as possible, and that's not always so easy to figure out.
Now, so your lab is working on quite a large number of projects currently, but is there anything else outside that you perhaps like to add to the mix? If you could choose anything, funding and expertise aside.
Oh, I think there are lots. I mean, lots of fields. I mean, sure. I'm not sure where to start. I mean, in terms of bioengineering, I mean, you mentioned artificial intelligence. I think the whole area of immunology, which has, many dimensions. I mean, I think there are many, many areas, and some of them don't have labels, and people are going to think of new ideas. I just think there's almost anything that can advance our understanding or create new tools to do good things or to come up with treatments would be good.
We've certainly seen some quite dramatic results recently in trials of combination drugs for melanoma, which certainly made headlines in the UK. Are you approached by any groups who have a problem? For example, I know you have a lot of partnerships with other labs, but how does that work? Are you approached by a group who has a problem to solve, or do you go to them and say, we could solve your problem?
Well, both happens, and I mean, like I said, we've certainly been approached by funding agencies, and like I mentioned, the Gates Foundation and Bill himself, and Juvenile Diabetes Foundation, Prostate Cancer Foundation, Helmsley Foundation, all of those came to see us, and there's some others as well. And then in terms of academic labs, we have people come all the time. I mean that. That has led to collaborations with different groups, like say at Johns Hopkins, at Mass General Hospital. And sometimes our students, see tools that other people have, like you mentioned, artificial intelligence. So we've been talking to people at MIT, like Regina Bargeli, who's expert at that. And so it happens all kinds of ways.
A quote that's been attributed to you is think big, don't give up. So I was wondering, is there anything that you think sounds big right now, but looking back in 10, 20, 50 years' time, you think we'll be glad we aimed for? And you can be very speculative here.
Well, I do feel that the next type of drugs are going to be these genetic drugs. I mean, I think these two areas and I've kind of mentioned them in different ways, but I do feel genetic medicine, which consists of siRNA or messenger RNA or different gene editing approaches. I think those are going to be very big as time goes on. And then I think cell therapy, which includes regenerative medicine. It also includes other kinds of things like different engineered cells. I think that those two areas, I think, will lead to all kinds of new therapies in the future. And they're starting to already, but there's just so much more that'll happen over the next many years, and so much more that needs to be done.
Professor Langer, thank you very much. It's been great to chat with you today.
Good luck on the podcast. Very, very good questions. You obviously did a good deal of research.